Hi everyone, hope all of you are doing well. Today, the topic which we are going to talk about is lipoproteins. So, we start with a small visualization. Okay, so imagine you've just enjoyed a delicious plate of Chole Bhature or maybe a rich Biryani. Your body has absorbed all those wonderful fats or lipids. Now, how do these fats, which are notoriously hydrophobic, yeah, that is the word, meaning they hate water more than a cat hates a bath, how do these travel through the bloodstream? Your blood is, for all intents and purposes, a watery highway. So we are talking about the blood. It's like trying to transport a truckload of ghee through river without a proper vessel. It's pure chaos, right? It would just clump up, causing blockages and biochemical disasters. So your body, in its infinite wisdom, has engineered an incredibly sophisticated solution. This sophisticated solution is basically lipoprotein. Think of them as the highly specialized lipid taxis or the molecular Dabba-Walas of the circulatory system. They are self-assembling, intelligent complexes designed to ferry precious fats like triglycerides for energy, cholesterol for cell building, precisely where they need to go, ensuring a smooth, clock-free ride. Without them, our cells would starve for essential building blocks. Even hormones could go haywire, and our very existence would be, well, a sticky mess, right? Because it's fat. So why does this matter? Because in our country, with our evolving diets and genetic predisposition, cardiovascular diseases, the diseases of the heart, is a growing epidemic. So understanding lipoproteins is about understanding the root cause of heart attacks and stroke. It's about saving lives one lipid particle at a time. So let's unlock the secrets of these lipid taxis. First thing we start with is the structure and components. At their heart, lipoproteins are macromolecules. They are macromolecular complexes. Their brilliant design allows them to solve our ghee in river problem. So let's look at the architecture. So when we talk about their architecture, they have a hydrophobic core. The hydrophobic core is the inner compartment completely shielded from water. So this is the hydrophobic core. It is the inner compartment which is completely shielded from water. It's packed with the truly water-hating lipids. Now, what are these water-hating lipids? Triglycerides and cholesterol esters. Triglycerides are your body's primary energy storage. Think of these as the Aloo parathas packed for lunch. And cholesterol esters are the storage form of cholesterol. It's more compact and hydrophobic than free cholesterol. The next part of the structure is the amphipathic shell. Now the amphipathic shell is the exterior. It is the outer layer. It's designed to interact with both the watery blood and the oily core. So this outer layer is the special part actually. It can interact with both blood and oil. That is why it is called amphipathic. Now, the lipids which constitute this layer are phospholipids, free cholesterol, and yes, these are the special ones in lipoproteins, that is the apolipoproteins. Now, phospholipids are the packaging material with a water-loving head facing outwards, and they have a fat-loving tail facing inwards. So you can imagine this internal part is their tail and this outer part is their head. This is how phospholipids appear, with having a head and a tail. Then free cholesterol sits comfortably within the phospholipid layer to maintain fluidity. Then comes the apoproteins. Now apoproteins, these are the true superheroes. Think of them as the drivers, GPS navigators, and ID cards of the taxis. Okay, so these are very specific for lipoproteins. They dictate where the taxi goes and what it drops off. So by this description, you can understand that they might be playing a pretty important role. Now I like to come to the ingredients. Okay, so let's break down the specific role of our biryani ingredients. Okay, a bit rich in fats, that's why I'm saying that. So the specific roles of the ingredients. First, we talk about triglycerides. Triglycerides are the fuel. They are used for energy storage. Then we have cholesterol esters. These are the cargo. They are stored in the core of LDL and HDL. Then we have free cholesterols. So free cholesterol is the structural support, right? Like we saw, it's crucial for cell membranes and it is crucial for making steroid hormones. Okay, so you might recall the various steroid hormones like testosterone, estrogen, and cortisol. These are made with the help of cholesterol. Then we have apolipoproteins. These are the VIPs. Now, why are they the VIPs? Like the main reasons why they are so important. Let's talk about these. So the first reason is they offer structural stability. Now, examples of the apoproteins which offer this are ApoB48 and ApoB100. So whenever we talk about apoproteins, we are going to see that they'll be termed in such a way. So you will get used to this terminology where they say ApoB or ApoE, etc. Okay. So the first one was structural stability. The second one is enzyme cofactors. They are the keys to operating certain enzymes. And now examples of these is ApoC2 and ApoA1. So ApoC2, what it does, it specifically activates lipoprotein lipase. Now I'm going to also give you a mnemonic soon about this, but just remember this for now, that it cuts the triglycerides to release energy. Okay, that is lipoprotein lipase. Then we have ApoA1. ApoA1 activates LCAT enzyme. It helps acquire cholesterol onto HDL. Then the final function I want to talk about here is as receptor ligands. So as receptor ligands, they are acting as the ID batches, like I told you. So examples of these are ApoE and ApoB100. They allow the particle to dock at the LDL receptor. So that is their function as a receptor ligand. So you can see here the three major functions of lipoproteins, which is structural stability and as enzyme cofactors and also as receptor ligands. Okay, so I hope the three major features or the functions of apoproteins are clear to you. Let's move on to something else. Let's take a look at throughout your body's transport system. What you will find is we have 5 major classes of lipoprotein. We are going to talk about the ones taking fat to your tissues and the ones bringing back the fat from the tissues to the liver. Now, amongst these classes, the first one is chylomicrons. So chylomicrons, you can think of them as the Samosa Delivery Van. They are made in the intestines, specifically the enterocytes of the intestines. After you eat, say, a fatty meal, okay? We were talking about biryani, right? The cargo here is 90% dietary triglycerides. So chylomicrons are the lipoproteins which carry triglycerides mainly, and 90% of their cargo is triglycerides. The driver or the surrounding element, the apoprotein in chylomicrons is ApoB48. This is unique to the gut. Then we come to the journey. So the journey is they enter the lymph, then the blood, and once they are in the blood, they borrow certain tools. Now what are these tools? Certain tools come up. These are ApoC2 and ApoE. Now they get these tools from HDL. You can see we are touching a bit upon the metabolism as well. Even though I don't want to touch too much on the metabolism just yet, but you can see how these things work. And finally, we come to the drop-off. They go past muscles and fat tissues, and ApoC2, this one which they acquired from HDL, it activates lipoprotein lipase, which is like the toll booth. So lipoprotein lipase strips the triglycerides out for energy or storage. Finally, the shrunken remnants go to the liver and they are recycled. This is why your blood plasma looks milky if you draw blood right after a heavy meal. That is why whenever you are going for a lipid profile test, it is advised that you at least fast for a period of 12 hours. Because chylomicrons are the major lipoproteins which are present in your blood right after a fatty meal. So now you can see this overall layout which we talked about. What we discussed here is, the first thing we discussed was the origin. Then we talked about the cargo. Then we talked about the driver. Then we talked about the journey. Then we talked about the transformation, and finally the destination or end, right? These 5 major subheadings will help us understand every single lipoprotein in a more simple way. So I'm going to try to incorporate this into all the different lipoproteins. Let's move on to the next lipoprotein. This is the VLDL. Okay, so the origin of VLDL is the liver. Where was the origin of chylomicrons? Yeah, the intestines. The liver exports fats it made from excess carbohydrates. So doctors tell you all the time to eat less rice or sweets, right? Because when there is excessive carbohydrates in your body or your system, the liver converts them into fats. So these fats are transported from the liver to the different tissues of your body through the VLDL. The VLDL is also called the very low density lipoprotein. The next we have is the cargo. The cargo is the endogenous triglycerides. What was the cargo in chylomicrons? It was exogenous triglyceride. The difference here is this is only 50 to 70%. So it's not 90% like it was in chylomicrons. Then we come to the driver. The driver is the apoprotein, right? So the ApoB100 is the driver here. It is also the big boss protein. It is the bigger protein amongst the apoproteins. And the journey is basically similar to chylomicrons. VLDL borrows ApoC2. So we discussed how HDL can offer ApoC2 and ApoE, right? HDL can offer ApoC2 even to VLDL. So VLDL borrows that. It drops off fat to various tissues. And finally, the transformation, as it loses all the fat and all the cargo, it shrinks and becomes denser. When it becomes denser, it transforms into something called IDL. This is also called intermediate density lipoprotein. We are not completely going into the depth of the metabolism of each lipoprotein here, but we are grazing the metabolisms here. So you will get a good idea about how the metabolisms work. Let's now move on to the next one. So we ended at the intermediate density lipoprotein, right? So let's discuss about the ideal. Now, the good thing here is we don't have to discuss much about the ideal because it is a transition particle. half goes back to the liver, and half gets further stripped down by an enzyme called HTGL to become the infamous LDL. So instead of going into the depth of IDL, we can see that VLDL forms IDL, and the major function of IDL is it's a transition particle. Half of it goes back to the liver, and the half of it can be broken down and converted into LDL, which is low density lipoprotein. You might have heard about the good cholesterol and bad cholesterol statements or anecdotes, right? So this is important for medical purposes. When we talk about the origin, it is formed from IDL in circulation, right? So the origin is IDL. Then the cargo is cholesterol. Mostly it is cholesterol, and the driver is ApoB100. So at the back of your mind, you remember that LDL is formed from IDL. So the driver is also related to IDL, which is ApoB100. And the main reason for the journey is to deliver cholesterol to cells for membrane repair and hormone synthesis. So the journey is pretty important actually for LDL because it is actually doing a very important job. Cells have an LDL receptor that grabs the ApoB100 and pulls the whole particle. So we saw how VLDL comes from the liver, it transforms into IDL, and IDL transforms into LDL. And LDL, inside, it's present in the blood, it goes to the various cells of the body. These are all the peripheral cells and tissues which need cholesterol. So we discussed how hormones need cholesterol and the membranes need cholesterol. And of course, after the cells have taken the LDL, the remaining goes back to the liver, and it is taken up by the liver as well. That is the entire journey, and I have covered the transformation and destination as well. So whenever we talk about LDL or HDL, the term bad comes up, right? Why is it called bad? So the answer is if you have too much LDL, or if it gets oxidized in your blood vessels, it crashes into your arterial walls, and this leads to something called atherosclerosis. That is the only reason why it is called bad. But even though it is called bad, it is very important for providing the cholesterol to various cells of the body. So it's not completely bad actually. And if you don't know its actual function and importance, you would really consider it to be bad. So being educated about LDL is pretty important so that you actually understand in what context we call it bad. Okay, so now that we have talked about the LDL, let's move on to the HDL, right? People usually talk about LDL and HDL in the same breath, so they have to be talked about together. So till now we have discussed the delivery trucks. These are all the delivery trucks, lipoproteins. Now we need to talk about waste management, okay? So when we are thinking about this in terms of like an analogy where the delivery trucks are delivering it to the various cells of the body. And then there's waste management, which does the opposite. And this is what HDL does. Just like we were talking about the origin of the different lipoproteins, we'll talk about the origin of HDL as well. It is liver and intestines. Now, this is a bit surprising, but it will get more clear if you read the metabolism, but I'll try to explain it to you more. Basically, the synthesis of HDL starts as a flat disc, and then it changes its shape in the body. And this formation mainly occurs in the liver and intestine. That is why that is the origin of HDL. The cargo is mostly cholesterol, and the key driver is ApoA1. And of course, the special part about HDL is there are several apoproteins connected to HDL, but ApoA1 is like the key driver. And the major journey or the mission of HDL is reverse cholesterol transport, or in short, RCT. This is the cleanup crew or the Swachh Bharat Abhiyan of your blood. HDL travels to tissues, picks up excess cholesterol that cells don't need, and ferries it back to the liver for excretion. So unlike all the other lipoproteins which we have discussed, they're taking the fats to the various tissues. But HDL is doing the opposite. It's taking the excessive fats and bringing it to the liver. So like always, I just want to end this with a summary. So the summary will have only the high yield takeaways. So the first one is Chylomicrons. Basically, they transport dietary fat. The major apoprotein is ApoB48, and the origin is the gut. Then there was VLDL, and it transports endogenous fat. The major apoprotein was ApoB100, and the origin was liver. Then LDL, we saw that it's also called bad cholesterol, but it's not completely bad, and it delivers cholesterol to the tissues. And the major one here was apoprotein, it was ApoB100. Of course, high levels of LDL can lead to atherosclerosis. Then we had HDL, which participated in reverse cholesterol transport. This is the only one which takes cholesterol from the peripheral tissues to the liver. And there are many apoproteins related to HDL, but the one I focused on is the ApoA1. The high levels of HDL are considered to be protective. And we talked about two enzymes. One was lipoprotein lipase, and the other one was lecithin cholesterol acyl transferase. So lipoprotein lipase cuts the fat, and the LCAT enzyme traps cholesterol. Here I wanted to give you a mnemonic. So C2 for cut it. Okay, so we saw how C2 is involved in activating lipoprotein lipase. So it sounds like cut it, so C2. And the mnemonic for A1, which is stimulating LCAT, A1 for acquiring. So it basically acquires, right? It traps the cholesterol. That is how you can remember it. So this is the first video in lipoprotein metabolism and chemistry. It basically gives you an intro into what lipoproteins are. This creates a good starting point for you to understand even more complex parts about lipoproteins. What you can do now is, even if you go into a book and study about this, you will understand what is going on because you understand what lipoproteins are really. In the next part, I plan to actually cover the metabolism and delve deeper into this. So thank you for watching. Hope you enjoyed this video and I can't wait to meet you in the next one.